plasma membrane and its component, and especially fluid mosaic model, which is given by Singer and Nicholson, right? Yeah, Singer and Nicholson ಬಗ್ಗೆ ಆದಷ್ಟು ಮುಗಿಸಿ we started actually the transportation ಹೇಗೆ ಆಗುತ್ತೆ ಎಲ್ಲಾನು ಕೂಡ ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದ್ವಿ. Okay, so in today's class we will first learn about transportation and then we will move on to the next one. Okay, good evening everyone who are in YouTube also. Okay, so we'll start the class then. So everyone is here. Okay, so I already muted everyone and started recording. So we'll see some questions first before we starting the classes. Okay, the first question is, which of the following statement is correct? Okay, they are asking which statement is correct. So first option is cell membrane is involved only in exosmosis. Cell membrane is involved in endosmosis. Cell membrane is involved in both exosmosis and endosmosis, and none of the above. Okay, so you can take your time and you can answer. So I will give you 5 seconds and yeah, got it. So I'm just launched the poll. Yeah, I launched the poll, guys. So I think probably you guys already got the answer. Who are in the Zoom and answer in the Zoom live chat, Zoom poll, and who are in the YouTube live can answer in the YouTube live chat. Okay, I'm waiting from everyone. Screen is not clear. Huh? It's clear, dude. I can see in YouTube. What's happening, Maisha? What kind of problem you are facing? Is any black or anything is there? Yeah, thank you. Yeah, exosmosis means the water going outside from inside. Okay, endosmosis means from outside to inside. Black, bro, do one thing. So once go out and join back again, Maish. Once go out of the class and join back. Okay, so in YouTube I'm not getting any answers. Good evening Gopika Anu. Okay, last 5 seconds, guys. 5 4 3 2 1 0. I'm ending the poll and I'm sharing the results. Good evening, Kirti. 10% chosen option A, 24% chosen option B, 66% chosen option C, and nobody chosen option D. Okay, exactly. So the cell membrane help in several processes. The one of that is the transportation. Okay, good evening, Raksha. The transportation, especially maybe water. That's what we call it as the osmosis, right? The transportation of water from inside to outside or outside to inside. When there is like change in tonicity, that means the concentration of solutes, excess water may move from outside to inside. That's what we call it as endosmosis. That means going inside. And when there is another changes, the water may come from inside to the outside. We call it as the exosmosis. Both of them actually happen through the plasma membrane. So the correct answer is involved in both endosmosis and the exosmosis. Okay, yeah, now we will move on to the good, Mahesh. Next we will move on to the second question, which is easy, and the question is long. Okay, take little time to read that question. The question is, keeping in view the fluid mosaic model for the structure of cell membrane, which of the following statement is correct with respect to the movement of lipids and proteins, okay, from one lipid monolayer to the other, okay, described as the flip-flop moment? CBSE, they asked in 2008. So just remember, CBSE, this question is from the CBSE only. Okay, the first option is, while protein can flip-flop, lipids cannot. Second option is, neither lipids nor proteins can flip-flop. Option C, both lipids and proteins can flip-flop. And option D is, while lipids can rarely flip-flop, proteins cannot. Okay, read the question, take 5 seconds. So I launch the poll in 5 seconds. Who are in the YouTube live can start your answers now only. Okay, yes, yeah, everyone started giving answer. I'm launching the poll, so you can answer in the poll. Okay, I'm getting answers. Still, you guys have like 40 seconds left. We discussed regarding that. And who are in YouTube, I mentioned behind my photo is model for the, 1 second, model for the structure. Okay, structure of cell membrane. Structure. Who are in the YouTube because you can't see. Okay, I'm ending the poll in 20 seconds. You still have 20 seconds, guys. You can answer. I know you guys can answer. Yeah, I'm ending the poll in 5 seconds. I'm ending the poll and I'm sharing the results. 15% chosen option A, 8% B, 15% C, 62% D. Good evening, Darshan. And in YouTube, everyone gave the answer 4. Okay, option D. So we'll see. So what we discussed actually, the lipids and proteins both will be present in the membrane lipid bilayer, right? This is one monolayer and this is another monolayer. So by joining it will become bilayer, right? We told the lipids have lateral movement and they also have the transverse movement. That's what we call it as the flip, or this side may be flop, right? But we mentioned that these proteins may have the lateral movement. That will be a slower lateral movement. There will be no flip-flop movement in the proteins. So you can arrange why the protein can flip-flop, lipids cannot. It's absolutely wrong. Neither lipids nor proteins, wrong, because lipids will do both lipids and proteins can flip-flop. It's wrong because proteins can't flip-flop. The correct answer is while lipids can rarely flip-flop, proteins cannot. Okay, why we told lipids can rarely flip-flop? Because it takes a lot of time for a lipid to transfer from one particular monolayer to the another monolayer. Okay, there are some other protein may help in doing that process. Okay, so correct answer is option 4. We will move on to this time. Types of transport we are discussing, right? Why is the flip-flop movement necessary? Good question, Shivan. Okay, show last slide. Last slide. So CBSE. No, this is in CBSE only, NCERT only. In CBSE, they mentioned that the lipid layer can show the flip-flop moment. They mentioned, but they didn't mention that protein won't show the flip-flop moment. Okay, they gave a half statement in CBSE textbook. Okay, what is the necessity for the flip-flop moment? Good question, Shivan. Okay, so what happens whenever a plasma membrane is like this? Okay, remember like this. Okay. This is think that this is one lipid monolayer and think that this is another monolipid bilayer. Then where these phospholipids are secreted is this these are secreted in here only. Manisha, wait for some time. Okay, so what do you guys think? These phospholipids and proteins are secreted in plasma membrane only? No, they normally secreted in the endoplasmic reticulum. Okay, this secreted phospholipid should transfer from here to here, right? Probably destination is outer membrane. You imagine like this. First, it will join to this particular membrane, inner membrane. Then, due to the activity, this will transfer to here. Okay, so that's the one reason to expand or to growth of the plasma membrane. There are some other reasons also there. The composition of lipid membranes in both ways should be maintained in a particular way. Okay, to maintaining that particular necessity or composition, they also do the flip-flop moment. Okay, the first question is this one, Manisha. Just take a screenshot so we can move on. Okay, yeah. So yeah, we are discussing about the. Good evening, Salma. Hi. So where is Ashwin? Probably Ashwin not in the class today. So types of the transport we are discussing. So I told you transport means movement, right? What will do the movement? Movement will happen of the different molecules, right? Different molecules or ions will happen. We can divide into two types. Passive transport, that means without wasting any energy, right? No energy needed for this kind of molecule. And another one is the active transport with energy. Okay, and all of this transport will happen through the help of the plasma membrane, or you can say cell membrane. PMCM means don't think the prime minister and chief minister. It's a plasma membrane and the cell membrane. Okay, don't think in that way, another way. Okay, so we will see about the passive transport then. Passive transport, the molecule will transfer from the higher concentration to lower concentration. Why it happen like this? Why they transfer from higher concentration to the lower concentration? Can anybody answer this question? In passive transportation, the molecular or ions transfer from higher concentration to lower concentration. Exactly. That's because no energy diffusion, maintain equilibrium. Simple. Exactly. Right. So wherever in the nature, you know about the thermodynamics and you know about the laws of the thermodynamics and all the things, right? So everything want to go for equilibrium state. So it will be confined to that. Okay. There will be no net movement, right? So what happens when there is a gradient is created? Okay. Gradient means what? Think that there are like this many compounds are here, and there is only one compound is here. So all these compounds having the more energy, right, compared to this one. So they want to transfer from here to here. That means from the higher concentration to the lower concentration. Exactly. That's what we call it as a gradient. Okay. We call it as the gradient. Okay. Today very excited to Venkat. Why is that? So up until what it will continue? It will continue until it will reach, or it will break the gradient, and it will reach a state, we call it as the equilibrium, right? It will reach the state of the equilibrium. Exactly. So you will learn about the equilibrium more in detail in the chemistry, right? What is the equilibrium and how it will be maintained, and what are the different kinds, all the different kinds of things. Okay. Think our cell is also in the same way. Okay, so whenever there is a high concentration and within the cell is a low concentration, or vice versa, the molecule wants to move from the one concentration to another concentration. Okay, that's actually by two different methods. One is the simple diffusion. Okay, one is the simple diffusion. What is the meaning of simple diffusion? There is no extra proteins are involved. Okay, simple diffusion means what? No extra proteins involved. No extra proteins involved here. Okay, so it's simple like how can I put it? So is like a simple diffusion means there is something is cooking in your kitchen, and the fragrance or the smell will transfer to whole house. Okay, here what's happening? The air current will transferring that particular molecules of that particular smell. Okay, nobody is transferring that, right? That's called simple diffusion. This will happen for the smaller molecules, how can I put it, non-polar and uncharged molecules. Remember, uncharged molecules. Where it will happen? It will happen to the very tiny and which are non-polar and which are uncharged molecule. This kind of simple diffusion will happen. Okay, so it may be a smaller kind of anything or all those things. Okay, so the second type is there, a facilitated diffusion. Okay, so what about other molecule? There may be some polar molecule, or there may be a molecule which is having some charge or anything. Then how it will pass? It have a facilitated diffusion. Facilitated means there is someone which is actually helping in the movement or helping in the movement. Okay, so it is again divided. It will happen through a protein. That's what we call it as. It will happen through a protein mediation. Okay, a protein will help the molecule transfer from the one media to another one. So in the protein there are like two types are there. One is the channel protein. Okay, channel protein, and another one is the carrier protein. So what's the difference then? Okay, as you can observe in this particular diagram, so molecule concentration is highest here. This particular molecule is transferring directly from here to here. Okay, it's showing a simple diffusion. Okay, yeah, no. That means the molecule which are showing the simple diffusion are smaller and non-polar molecules. Okay, so smaller non-polar uncharged molecule. Smaller non-polar uncharged molecule will pass down here. Okay, will pass. will pass. Okay, so there are some other concentration, but these molecule can't show this kind of diffusion. Probably this is a polar molecule. Consider it's a water molecule. Okay, then how it will pass? It have a protein. First protein is a channel protein. Channel protein will have a opening, a continuous opening in between them. So this continuous opening will help the molecule transfer from one side to another side by, oh, 1 second guys. Sorry. By transferring through this particular protein. Okay, so the transformation or transferring from one side to another side will happen through this. Okay, so transfer will happen. And do you really think it will allow all the molecule and all the things? No, it will only allow the selected molecules. Okay, this will only allow the selected molecules. How can I put it then? So I will write it in this way. So the channels is the proteins with hollow inner side, but they are not allow all molecules. They are selective in function. Okay, selective in function. That means they select one particular one and they will transfer that. Okay, and second one, there is a called carriers. Okay, I will write both of the names here only. This is the channel protein. Okay, channel protein, and this is the carrier. Look here, the carrier is not like a channel. Okay, can it regulate the movement of the molecule? Yes, it can regulate. Okay, regulate means it can help that it will only select a particular kind of molecule and it will allow it. Okay, until I will tell you, until the equilibrium. And here, as you can see a carrier molecule have some modification. That means this carrier molecule will change its shape to transfer the molecule from one side to the another side. Okay, how can you differentiate between carrier and channel? Channel well won't go for any structural differentiation or structural modification, but the carrier molecule will go for some structural modification, and from how they will transfer certain molecules. Okay. The difference is, I will write down 1 second. Yeah, go for the structural, STR means structural modification. Go for the structural modification. Okay, by structural modification, they will transfer from the one side to the another side. Okay, remember, they may go in the simple diffusion or a facilitated diffusion. In the facilitated diffusion, it may be channel or the carrier. The very important thing you have to remember is no energy is involved in here. Okay, first thing is no energy is involved in here. Okay, then what's the important thing? They're always going according to the gradient. That means from the high concentration to the lower concentration only going up to what? Until reach equilibrium, until they will reach the equilibrium. Okay, until they will reach the equilibrium. Remember, so these simple kind of or passive transport mechanism may happen directly through the simple diffusion, or may happen through a channel or carrier protein. That's what we call it as facilitated diffusion. That means some other compounds, other structure is helping in the transportation, but whatever may be, here in the passive transport, no energy is involved. Okay, no energy is involved, and it will happen through the gradient only. Okay, through the gradient only until it will reach the equilibrium. Okay, that is really important. Okay, what's the difference between the simple diffusion and the channel? Exceptions of energy. Remember, in simple diffusion, there is no protein is involved, right? So I will write down here only. Okay, no proteins are involved. No protein involved. No protein involved. But if you take a facilitated diffusion, in this, specialized proteins are involved. Right, that is the major difference between the simple and the facilitated. Specialized proteins are involved. Okay, so it can't regulate the proteins, sorry, it can't regulate the molecules if there is gradient is very high gradient is there. One side 100 molecules are there, one is only one molecule is there, then it can't do anything. The molecule will pass down. Okay, but once the equilibrium is reached 50-50 molecules, okay, then everything will be stops there. Okay, the movement will be, net movement will be zero. Okay, that's facilitated. What is gradient? Gradient means the difference in the concentration, difference in concentration. 55% and 10%, that's a difference in concentration, right? That is the gradient. Does facilitated diffusion has polar and charged molecule? Yes, the facilitated diffusion normally may allow the polar and the charged molecule. Okay, while transferring the charged molecule, it will make it combine with some other molecule and it will transfer it. Okay, normally the charged molecule can't go through easily because it have the charge, right? It will start to bind to something. That's why. Sir, how channel protein stops certain molecules from going inside its continuous channel with no? That's what I told Jayant. So it don't have any ability to stop somewhere. Okay, it will only stop. Net movement will be stopped when equilibrium stopped. What is the meaning of net movement? Okay, so that's very important. Okay, the difference is there between the movement and the net movement. Okay, so I will take another one page. Try it just to show it. Okay, 1 second, guys. This chat box is okay. So what is the difference between a movement and a net movement? Okay, we will think about a membrane here. We'll think this is very really important concept. Okay, so think about a system here. Okay, a system here. There are like 5 molecules are here. Here only one molecule is here. Okay. So when this one molecule is transferring here because it wants to reach the equilibrium, so then you can call it as the movement because transferring from one side to another, from A to B, right? Happening from A to B is happening, right? So this is the movement. What is the net movement? One molecule is the net movement. Okay. Then we will take the second case. Second case, what happened? A and B. So here 3 molecules are here, here 3 molecules are here. It's in equilibrium, but one molecule is transferring here to here. At the same time, another molecule is transferring from here to here. And that time, moment is there or not? Exactly. First scenario, second scenario is also moment is there, right? Transferring to A to B and B to A is happening. Moment is there. Is there any net moment is there? No. Zero net moment in the second one. Zero net moment. Exactly. What's the moment meaning of net moment then? Net moment means when you consider the complete movement from one side to another side and you minus it, if you get the zero, then the net moment means overall movement, right? So in second scenario, in the scenarios like this, in the equilibrium states, remember the movements will be there. Okay, the molecule move from one side to another side and this side, it will happen always. But there is no net moment. That means there is no change in the moment. Whatever the moment will be, it will be always in the equilibrium. This is the basic things to understand the equilibrium. Okay, remember in equilibrium, there will be the moment and there will be no net moment. Okay. It means, sir, both sides it will remain exactly. Both sides it will remain 3 only, even though moment is there. Overall concentration after, yeah, exactly, Soumya. Okay. Indian water into cell, but no net moment is affect water in or out. Exactly, exactly. Okay. So we will learn about more regarding these things one more time. Okay. So these are the things. There is no net moment. Once equilibrium reached, there is no net moment. Okay. Then there is active transport. There are some compounds which are larger in structure. Okay. And another one thing, they can't pass through this particular state. Okay. Through any kind of the molecules. And there are another one thing is there, a cell may be in a different condition. We will consider a cell is like this. It have 3 compounds inside, sorry, 5 compounds inside. And outside there is only 1 compound is here. But the cell needs this one compound also. Okay. But this is completely against the equilibrium, right? It's completely against thermodynamic. In this kind, what is the case? It's completely against thermodynamic rule. That means it's not going from higher to lower, it's going from lower to higher, right? That time, the plant cell utilize the energy in the form of ATP and it will transport inside. Okay, so active transport is against the gradient. It used for against the, I will write down below. Okay, I will write what's the meaning of this one, active transport. Transportation using energy. Transportation using energy, you can call it as the active transport. Okay, active transport maybe happen when probably gradient is against. Okay, against gradient. What is the meaning of gradient? As I told you, probably from the lower concentration to the higher concentration. Okay, yeah, low to high against gradient. I will write down in the bracket in here on. Okay, low to high. Or there may be another condition is here. The condition may be the active transportation is very quick. Okay, it's one of the like using one of the speed or quickest kind of thing. The cell may need molecules in more in less time. Okay, it may need the particular compounds in a less time. In their active transportation will happen. Okay, so active transportation one of the important thing in our body because it only maintain different concentration gradient. Okay, so normally when we are discussing about the channelization of neurons and whatever we are discussing, whatever I'm talking and whatever I'm doing, it's all because of the different signals which is passing in our body, right? How it is happening? All of this happening either through the different in concentration of the potassium and the sodium. So if you take your neurons, okay, think that this is a neuron. Okay, we'll simply write it in this way. Neurons. In neurons, what happens? Neurons in a regular state, the potassium ion will be increased inside and the sodium ion will be increased outside. Okay, so how this concentration is maintained? Through the active transportation process. Okay, through the active transportation process. And how can you move your muscles? How can you move your muscles? Okay, I'll write a kind of a muscle. Okay, because when calcium is channelized into your body, your muscle will show the movement. Okay, or the muscle movement. That's how it's happening. So all the concentration of different concentration or movement of the ions, we need active transport. Okay. How can a particular cell concentrate a chemical inside its cell? Because with help of the active transportation system. Okay. How can the plants will take the minerals, very rare minerals which is present in the soil by the process of the active transport. Okay. Then while running in a great speed, active transportation, no, not only running in a great speed, every time active transportation is happening. Okay, and active transportation make use ATP for the energy. Okay, how much ATP it will use? Okay, how much ATP it will use? The ATP, if you take your brain cell, almost like 70% of the energy used by the cells, okay, is used for the transportation, for the active transportation. But if we take your muscle cells, almost like 30 to 50% of the energy used in the cell actually used in the active transportation. Okay, so exactly this also happens through proteins. Okay, proteins. The proteins are different. Proteins are there, and remember the active transportation are highly specific. Okay, they are not pass all the things. Okay, something is there, I will pass. There is some other thing is there, they won't pass. Okay, they are specific to a particular compound. Okay, they are specific to a particular kind of the compounds also. Okay, what happens if there is any chemical? It will stop this kind of transportation. Yeah, there are some compounds. These are called inhibitors. Okay, inhibitors. What I can say is like inhibitors can stop this process. Inhibitors can stop the process. stop this process. That means there are some toxins are there. Okay, the toxin actually bind to this protein molecule and it will stop the active transportation process. So that's why that particular organism may die. Okay, or the cell may die. We can take an example of Mamba snake. Okay, Mamba snake. Okay, or you can take the example of some kind of toxins which is actually secreted in the this one scorpions. Okay, northeastern scorpion. All those scorpion venom or that Mamba venom. Okay, what it will do? This will go and attach to that protein and it will stop the active transport. Because of the stop of active transportation system, the person may die. Okay, there are some proteins also, there are some toxins also present in the plants. Those are called cardiotoxins, cardiotoxics. Okay, because once you ingest a particular chemical, the person will die because of the heart stop. Okay, why that will heart will stop? For the movement of the heart, we need a proper amount of calcium, sodium, and potassium ion concentration. When active transportation will be stopped, then heart will stop beating. Okay, what is the mode of transportation in active transportation? Through the protein. I wrote down here, black mamba. Exactly. Okay, so remember this thing, inhibitors can stop this process in that chip, in the carrier protein. Carrier protein, we simply shown it like this because to understand this, the protein structures will be completely different. Okay, can we produce the poisons? Yes, you can produce the poison, no problem. Yeah, we can move on then. These are the related things which are related to plasma membrane and the transportation system. Okay, next will be the cell wall. Cell wall is the important thing which can be present in the plants and fungi, not all fungi, in most of the fungi, and also in some protistants. Okay. Mohan, it happens through the protein only. I mentioned it here, right? It happens through the proteins. Okay. It may be a carrier, different types of carriers are there. That's why I didn't mention. That's not in the syllabus. Okay. So ion gated channels are there, carriers are there. It will be there. Okay. Yeah. In protistants. Actually, we are not mentioning it here because we are normally studying about eukaryotes, right? So I'm not mentioning it here. Okay. Some protistants. This have large pore. This one protistants. Okay, what is cell wall made up of? Cell wall is normally a rigid layer. Okay, cell wall is normally a rigid layer. How it will be helpful? How it will be helpful? This layer actually help in protection against mechanical and chemical damage. Protection against mechanical and chemical damage. Okay, mechanical means if you mechanically if you hurt something, a plant cell or anything, or chemically if you put some chemical, okay, by that it will be helpful. Then what's the composition? It will be vary. Okay, if I take three organisms, protistants we normally leave because it have a different variety of the plant cell wall composition. Important two things are plants and the second thing is the fungi. Right, in fungi the cell wall is majorly made up of a chitin kind of structure. So we mention it chitin only because chitin kind of a structure. It's both are we will consider it similar. Okay, and in plants it normally made up of the cellulose. Exactly, cellulose is a kind of carbohydrate, as you can think. Okay, it's a polymer. Okay, so what cellulose will do? This cellulose is one of the strongest compound in the nature. Okay, it make a large chain. Okay, and it will arrange itself to more the cell wall. Is it cellulose only present in there? No, it also have hemicellulose. Okay, and also the pectin. Okay, there are some other compounds also there. These are the major compounds. Okay, if you go for the plant cell wall, the plant cell wall will be, one second guys. Okay, function is here. Okay, the plant cell wall will be arranged. Mannose will be there. Yes, exactly, mannose will be there. And major these four in the primary. Okay, I will mention about the secondary later on. The plant cell wall will be formed outside that and it will form a structure for that particular cell. Okay, I will use some red color for the cell wall. Okay, think a white color is the plasma membrane and the red color is the cell wall. Okay, red color is the cell wall, think like that. And it will be deposited outside of that, and it will help in the protection towards the different kinds, and it will arrange it in a different manners also. Okay. So this is the structure, and this is the composition of the plant cell wall and the fungal cell wall also. Okay. So we will see something else also. So this particular cell wall, we normally call it as the primary cell wall. Okay, primary cell wall. Primary cell wall means a cell wall which is actually started in the initial days, or the primary one. Okay. So what happens when the particular plant cell is going through the aging process? Then it will secrete some second layer of the cell wall inside to the primary cell wall. That is what we call it as the secondary cell wall. Okay, secondary cell wall. Each secondary cell wall is different from the primary cell wall. Exactly. The primary cell wall normally contain hemicellulose more, cellulose and pectin and lignin. Okay. But if you compare the secondary cell wall contain cellulose, exactly. Okay. But less hemicellulose, but more lignin. Okay. It contains another one compound called lignin. Lignin is more stronger than the cellulose. Okay. So why it used another one compound instead of cell wall? You know that cells are growing, plants are growing. Okay. Cells becoming older means the plant is becoming bigger, right? Plant is becoming bigger means the plant cell will have more pressure to hold all those things, right? So to hold or to get more strength, it changed its composition from cellulose, hemicellulose, pectin, mannose layer to the cellulose lignin kind of layer. Okay. This secondary cell wall may be several layers. Okay. So for the several layers, we gave the name something like S1, S2, S3, S4 until Sx, several. Okay. That means secondary layer 1, secondary cell wall 2, secondary cell wall 3, and secondary cell wall 4, like that. Okay. In an extreme situation, okay, I will write down the plasma membrane inside only without any changes. Okay. In an extreme kind of a situation, there is another one layer will form. Okay. Remember something like this. Inside to this is called tertiary layer. Okay. Tertiary layer. Okay. What is the meaning of tertiary layer? Tertiary layer is normally present in more like secondary cell wall only, the plant will start to die. Okay, the cell will start to die. But in tertiary layer will be present in some of them. These have cellulose, yeah, base composition is same only, cellulose only, lignin, and also another composition called suberin. Okay, suberin. This suberin is actually compound of alcoholic nature. Okay, this lignin and suberin will stop the water move from one side to another side. Okay, this act like a completely barrier to the water. Okay, barrier to the water, but they will give great strength. Okay, where you can find a tertiary layer, the plants or the cell which are in term of death or in the senescence. Senescence means when it will lose all its leaf, right? On that kind of the time, or near to the death of that particular cell, you can find tertiary layer. Okay. So plasma membrane is outside or inside? You guys are asking. Exactly. Plasma membrane is always inside. Okay. Plasma membrane is always inside. Here only, plasma membrane. Okay. These primary, secondary, and tertiary layers pushing the plasma membrane inside. Okay. If plasma membrane is present outside of the, because you know that cell wall can't produce any more outside, because cell wall is almost like a non-living, right? It's not a plasma membrane to secrete something. Whatever secretion is happening from the cell membrane only or plasma membrane only. So the plasma membrane always reside inside. Remember, don't miss the terms. Okay. I'll write the diagram in another one section, something like this. Outer layer, next layer will be, we used blue color, right? Second layer will be the secondary layer, and the third one will be the tertiary layer, and the last one will be the plasma membrane, right? Plasma membrane, primary layer, secondary layer, and tertiary layer. Okay, inside to inside it will happen. Okay, primary layer will be initial in every organism, later secondary, later tertiary will come. So this is the arrangement. When you look at the two different cells, Okay, when you look at the two different cell, how it will be joined? Probably Shailaja asked the question in the last class, I think. Okay, so think that this is the one of the plant cell, and this is the one of the plant cell. Cell wall, I'm writing the cell wall. Okay, do you guys think anything present in between? Yes, there is a layer present in between. That is what we call it as middle lamella. Okay, middle lamella. Middle means what? Middle means in between. Lamella means layer, right? Middle means in between, and lamella means layer. So what is the meaning of this? This particular layer present in between the two cell wall or two cells. Okay, cell 1 and cell 2. Don't think that middle lamella present between cell wall and the plasma membrane. No, that's not correct. You can see here only. Plasma membrane is here of the first cell. Cell wall is here, primary cell wall, right? Middle lamella is here. After the middle lamella, you can find the another one cell wall and another one of the plasma membrane of the other cells. Okay. Correct, correct. In animals, in cell components exert less pressure. It needs less strength because animals won't grow like that much big, right? That's why. But we have the endoskeleton and all the things to counteract the pressure. Yeah. So middle lamella will be present in between the two cells. Middle lamella will be present where it will be present in between two cells. Write down the notes, guys. Otherwise, you will confuse with these things in between two cells. Okay. Then what are these middle lamella normally made up of the pectates. Okay, pectates of the calcium plus magnesium. Normally, it is normally made up of the calcium and the calcium and the magnesium pectates. Okay, so we don't have cell wall exactly. Okay, so these are the made up of the pectates of the calcium and the magnesium. So these are act like a binding. Okay, binding like a gum between one cell to the another cell. Is there anything between one or another cell? There is a small gap is there. That's we call it as the plasmodesmata. Okay, plasmodesmata. What is the meaning of plasmodesmata then? And formation of secondary cell will happen altogether, or will it happen cell by cell? Normally, cell by, I didn't understand exactly the question, Manvita. The formation of secondary wall normally based on that particular cell. Okay, normally based on that particular cell and the situation is going on. If it is going through senescence or it's transferring from primary xylem to the second, is it transferring to the xylem? All those will matter. Normally, fibers will having the secondary cell wall or tertiary cell wall. Okay. So as you can see here, in between two cells, a channel-like kind will be there. Okay, this channel will help in the transportation of material from one cell to the another cell. Okay. Primary cell wall will be there. Above that, below that secondary cell wall and tertiary will come. Okay, where pectates are present in the middle lamella. Cell wall only. Plasmodesmata, you look like a this one hole, right? Yeah, it's a channel. So in between two cells, you can find a channel kind of the structure. That's what we call it as the plasmodesmata. Okay, what is plasmodesmata? The plasmodesmata is the hole, or you can call it as a channel between or a connection link. How can we put it this? Okay, what's the best word you guys think? Okay, what's the best word you guys think? Connects to cytoplasm. Yeah, that's 100% correct. Okay, plasmodesmata, we can put it something like that. This is the bridge between two adjacent cell represented by minute pores. Represented by minute pores. That means you can call it as a hole also. Okay. No issue, Pawan. I didn't saw it. Okay. So in plasmodesmata, the plasma membrane of one cell to another cell is connected. Okay. Plasma membrane is also connected, and the cytoplasm is connected through here. Okay. The cytoplasm is connected. is connected from one cell to another through the process of plasmodesmata. Okay, so using the plasmodesmata, some of the molecule may move from one cell to another cell. This kind of movement we call it as the symplast movement. Okay, symplast movement. What's the meaning of symplast movement? The movement of molecule from one cell to another cell through the plasmodesmata, we call it as the symplast. No, none of this is real photos. These are actually computerized photo. Okay, computer-wise made a photo. Okay. So symplast. Sometimes this cells won't move along the cells. They actually go outside of the cell. This is what called as the apoplast. Okay. If the molecules pass through the plasmodesmata within the cell from one cell to another cell, we call it as the symplast. If the molecules do not enter the cell, it is going in between space or space in between two cells. Like there are like, if the space is like this, a space will be there right in between. If the molecule passing through this space, then we call it as the apoplast. Okay. Can a cell transfer its fall to another? No, nothing. Cells never think like that. Okay. So this is also not a real photo. All is are computerized photos only. Okay, so this is which related to cell wall and its function. Okay, one question came, you answer the question. Meantime, if you have any doubts, you can ask. Okay, which of the following structure between two adjacent cells is an effective transport pathway? Plasmodesmata, plastoquinons, endoplasmic reticulum, plasma lemma. Okay, I'm launching the poll without wasting any time because I think you guys can answer directly. Okay. Okay. I'm waiting in the YouTube also and in here also for everyone to answer. Okay, then you guys still have 30 seconds. Then I'm closing the polls. So think about one more time. Apoplast movement by plasmodesmata. No, apoplast movement is not from the plasmodesmata. If it is from plasmodesmata, this is symplast. If it is not from the plasmodesmata, then it's apoplast. Show the last. I will show this last slide for me. Sorry, Maulya. Once previous. One second. Once I complete this question, then I will go back. Okay. Yeah, I'm ending the poll, guys. Then I'm sharing the results. 83% chosen option A, 2% B, 13% C, and 3% D. Exactly. And even YouTube, everyone chosen option A. Okay. Correct answer is yes. Plasmodesmata is the correct answer. Plastoquinons are a kind of chemical compounds which is present in the plants. Endoplasmic reticulum is an organelle, and plasma lemma is the plasma membrane name only. Okay. Somebody wanted to see last slide. I'm just passing it here. Okay. Remember, guys, here along with the plasmodesmata, sometimes the endoplasmic reticulum also transfer its tubules from one cell to the another cell. Okay, endoplasmic reticulum, but we won't consider that. We won't consider that as a plasmodesmata. Plasmodesmata will be there. The endoplasmic reticulum utilize that plasmodesmata and transfer its tubules to the another cell to transfer some of its material to the another cell. Okay. In a passive transport, facilitated diffusion use what kind of protein? What kind of protein? I didn't understand. Facilitated, it means carrier protein and channel protein. Carrier protein and channel protein. Okay. I think we can move on to the next slide. Okay. Next one is the endomembrane system. This is really important. What's the meaning of endomembrane system? So now we are going to study about actual organelles. Okay. But there are group of organelles are there. Group of organelles are there. What they will do, they are connected from one another and they will show a coordinated movement. Okay. Yeah. All of them are membrane bound only. Remember, all of them are membrane bound. Okay. All of them are membrane bound, especially they are single membrane bound. Okay. Single membrane bound. Plasmodesmata. Good question, Chirant. Chirant is asking, the plant cell only have the plasmodesmata, animal cell don't have the plasmodesmata. Then what happens in what happens the moment in the animal cells? Animal cells have some other kinds of the things called a tight junction, gap junctions, okay, zigzag junctions. There are like different kinds of junctions are there. Through which actually transferring of materials will be happen. Okay, we'll see that in the structural organization. Passing facility inside membrane. Okay. So these are all of this endomembrane organelles are membrane bound, especially they are single membrane bound. Okay, but they are show the coordinated functions or coordinated movements. That means everything will be adjusted to one and another. Okay, it contains only 4 organelles. Remember, that will be endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Okay, remember only 4 of them are related. Okay, you guys can ask me how it's related then. Okay, in a simple story, I can tell you the endoplasmic reticulum is the base which actually enclosed, or it will end up to this nuclear membrane. Okay, this endoplasmic reticulum gives rise to some of the vesicles and all the things from where Golgi complex will born. From the molecules of Golgi complex, the lysosomes will born, and this moment of or the distribution of the things from the endoplasmic reticulum and Golgi complex, the vacuoles will born. Okay, everything is interconnected. If you remove the endoplasmic reticulum, you can't find other 3 things. Okay, that's how interconnected it is. Okay, is plasmodesmata and synapse function is same function. Synapse, if you are talking about this one, what is that? Neural tissues is different. Okay, synapse will be different, and this is different because in synapse the movement will be happens through channel or carrier proteins through a chemicals. Plasmodesmata is a direct connected thing. Okay, so remember guys, endomembrane system contains only 4 organelles: endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Why we call it as endomembrane systems? Because these are show coordinated functions or coordinated movements. Okay, that is very important. Bound or bond, membrane bound. These are single membrane bound. Single membrane bound. Single membrane bound. That means it will have a single membrane outside. Single membrane bound. Okay. Yeah, we will move on to the first one. That's called endoplasmic reticulum, or ER. Okay, ER is the one of the super important organelle within our body. Okay, so why it is so important then? Because it will make a lot of functions in your cells, which is actually the result of your body in a proper activation. Okay. Golgi complex is work along with the endoplasmic reticulum, and the materials will be normally given by the endoplasmic reticulum only. The materials which is made up of Golgi. That's why we call it. Sometimes we mention it as like Golgi is born out of the endoplasmic. Okay, so it's like kind of like saying only. Okay, so endoplasmic reticulum is bound to a nuclear membrane. Okay, so what are the other characters? This can extend up to end of cell. Remember, not in all the case, it can. Okay, it can extend up to the end of cell. May pass through plasmodesmata also. It may pass through the plasmodesmata also for connecting to the other cells. Okay, what are some other things then? Can extend up to, okay, no issue. Okay, can extend up to that range. Then what are some other? You can find 2 different kinds of the reticulum. Endoplasmic reticulum is there. One is the smooth endoplasmic reticulum, and another one is the rough endoplasmic reticulum. Okay, smooth ER. Second one is the rough ER. Why we call it in this way? This rough endoplasmic reticulum is bounded by ribosomes. ribosomes, yes, exactly. I can write in that way only. Yeah, ribosomes present on surface. Then you can call it as rough. So it will be not like a regular, right? It will be somewhat like it's not a smooth surface, it's a rough surface. But smooth endoplasmic reticulum do not have ribosomes attached to it. Okay, so how can you find it? Either rough endoplasmic reticulum, the ribosomes will be attached, but in smooth endoplasmic reticulum, it's completely smooth. Okay, there is no ribosomes, nothing. Okay, it's simple. So as you can see here, right? So some of the ribosomes, some of the endoplasmic reticulum have ribosomes. So you can call it as the RER, as you can see here. But some of them don't. You call it as the SER. Okay. If you compare this one, sir, does the function is same in both? I will go through the function in the next slide. Okay. So when you go for structures, ribosomes is currently positive structure on so when you go for its structures, it actually found in 3 different types. Okay. The first one is cisternae. Okay. Cisternae is given that ribosomes are stick to plasma membrane. No, not to plasma membrane. It's a membrane, right? We normally won't call it as a plasma membrane. We call it as the ER membrane. Okay, endoplasmic reticulum membrane. Okay, yes, ribosomes are stick to the endoplasmic reticulum membrane. Okay, there are 3 different types. You can make it in this endoplasmic reticulum. One is the cisternae, and another one is the tubules, and last one is the vesicles. Okay, what are 3 different structures then? The cisternae means something like this. Okay, something like this is called cisternae, a kind of flat structures which is present one and another. Okay, that one, Acharant. I will come to that. And tubules means a tube-like structure. Okay, a tube-like structure. Vesicle means a circular structure. Okay, vesicle, a spear-like structure. A tube-like structure, a spear-like structure, and the last one, cisternae means cisternae is a pad-like structure. You can say flat structure. Okay, like in you can compare it like that. Okay, exactly. In here, the ribosomes are attached to the endoplasmic reticulum, and if you go for prokaryotes, the ribosomes may attach to the plasma membrane also sometime. Okay, that's there. Probably I left the point in there. Okay, no issue. Show that in a photo. Okay, I will do that. So here, this is not exactly clear. Okay, in this particular picture, I try to show that. So these are the tubules. These are the tubules, as you can see here, and these things are the cisternae. Okay, these things are the cisternae. Vesicles, they didn't mention it. Okay, vesicles, they didn't mention it. This will be somewhat like this. Okay, this will be somewhat like this. So this will be vesicles. Okay, vesicle they didn't mention in this particular photo, but you can see the cisternae and the tubules. Okay, this is how it will be formed. Okay, these are the major 3 different structures which is present in the endoplasmic reticulum. When you go for the function, okay, we will go for the functions of the 2 different kinds. First one we will encounter with the smooth endoplasmic reticulum. So what is the important function of the smooth endoplasmic reticulum then? Okay, the smooth endoplasmic reticulum, the first function is it is taking care of the lipid synthesis. Exactly, someone answered before me. Lipid synthesis, that means synthesizing the lipids. Okay, that is the important thing which related to the smooth endoplasmic reticulum. Okay, and in the muscle tissues, if I take an example of muscle tissue, it have another one function, that is the concentrating or to holding, I can say. Okay, in this one, how can I say? In the muscle tissues, it will hold calcium ions, Ca2 plus ions, okay, which actually help in the muscle contraction. Calcium ion help in the muscle contraction. And the products which is synthesized by the smooth endoplasmic reticulum also help in the building of the Golgi complexes. That means the products are base for Golgi. Okay, what are the products it's synthesizing, right? It may be lipids or it may be some other compounds which actually help in the building of the Golgi or to join the Golgi. Okay. Yeah, synthetic compound or synthetic lipid means the lipids which is synthesized in biomolecules. We will see, okay, what is the synthetic lipid and derived lipid, all those things. Synthetic lipids means the derived lipids, what you are taking here, everything will go through the smooth endoplasmic reticulum to the Golgi. Okay. Next one is the rough endoplasmic reticulum. Exactly. The important thing is the protein synthesis. How it will do the protein synthesis? We already mentioned ribosome will do the protein synthesis, right? Yeah, ribosome will do the protein synthesis. The structure will somewhat like this. Okay, I'll write down here. The rough think that this is the rough endoplasmic reticulum. Okay, as you can see, simple hole, right? Here, our ribosomes will be attached. Okay, here our ribosome will attach, and what it will do, ribosome will do this mRNA, take this mRNA and transfer this into the protein. I will take a red color protein. This protein will be start embedded within the rough endoplasmic reticulum tubule. Okay. How? Yeah, Jeevan. SCR also help in the formation. That's why, like, lipid synthesis means the lipids are present in the cell wall and plasma membrane and all the things, right? It will help in the cell membrane or plasma membrane formation. Yeah. It helps in the protein synthesis by accumulating different kinds of the ribosomes on the surface, and it also helps in the protein modification. Okay, protein modification means the proteins are not functional when it's formed. Okay, it have to go through different structural changes, modification, then only that will become that action that actually happened in the protein here. Okay, and what is the another one thing? Glycosylation or adding glycosylation is the better thing, I think. Okay, glycosylation happens here. Glycosylation initiates here. Initiate here. What is the meaning of here? Glycosylation means what? Glycosylation means adding carbohydrate group to protein. Adding carbohydrate group to protein. So does all type of cell contain this? Yes. All eukaryotic cell will contain smooth endoplasmic reticulum and rough endoplasmic reticulum, but the composition will vary depending upon the function. Okay. Yeah. Any other things? This also help in production of several hormones. Okay. Hormone production, and some other lipid kind of production also. Okay, that's also very, very important thing which related to this. Actually, I left one more thing, guys. Sorry for that. Smooth endoplasmic reticulum function, important function, removing toxic molecules. This is very important. Okay, so toxic molecule. Whatever the chemicals you are taking, either you may be taking antibiotic, right? Antibiotic is harmful for the bacteria, but this is not the compound which normally present in our body, right? So those antibiotic destruction of the antibiotic or removal of the antibiotic will happen in the smooth endoplasmic reticulum, and exactly same kind. So destruction or removing of the alcohol from your blood also happen in the smooth endoplasmic reticulum. Okay. So it have two functions. One is the lipid biosynthesis, and second one is the removal of the toxic molecule. Then where it will be present, smooth endoplasmic reticulum, you can find more in liver cells. Okay, liver is the place which actually contain the molecule, right, lipid, and it will help in the transformation. So you can find more smooth endoplasmic reticulum in the liver, because the liver is the site where actually lipid synthesis and removing of the toxic molecule will be happening. Okay, that's why who are fat, okay, that means who are having obesity or having more weight, if they started drinking, their liver may go for cirrhosis. Okay, that means a problematic condition of the liver. Why? Because there are like a lot of pressure will be put on the liver, because the SCR can do a certain function only at a time. It have a multiple function, because it have to redistribute alcohol, it have to again go for lipid synthesis, then it will go for higher pressure, then it will be problematic. Okay, both contribute to formation of the plasma. Exactly, both will contribute to the formation of the plasma membrane and also in the formation of the other molecules. Okay, so all the cells will have this smooth and rough endoplasmic reticulum, but the composition will be vary. Okay, if you take the liver, smooth endoplasmic reticulum will be more. If you take a muscle, so then also smooth endoplasmic reticulum may be more. Okay, where actually protein composition will be more needed. Okay, that means if you take testicles, okay, the cells, testicular cells, their rough endoplasmic reticulum will be more. Okay, because it have to produce the hormones, right? Because of that function. Sir, then bile is produced in the RE. Exactly, bile is produced by the endoplasmic reticulum only, but we simply call it as from the cell wall. So that's why it happens, right? When I'm talking about testosterone, I will tell you, like, for the smaller children, I will tell you that males will produce the testosterone. Then I will tell you that testicle will produce the testosterone. Okay. Then I will tell something like, there are like interstitial cells will be there. Those cells will produce the testosterone. Okay. Then you go for cytology inside the cell. Then you will say that rough endoplasmic reticulum will produce the testosterone. Okay. Everything is same, but we are watching from the different levels. Okay. Our where you are watching the perspective, the things will be different. Okay. But you have to imagine about these things. Okay. How it's working and all the things. Whatever the products may be, the cell products may be, or tissue products may be, that may be synthesized from the endoplasmic reticulum or from the Golgi or any this one, plastid or something. Okay, we all move on then. Second one is a Golgi. Okay, Golgi is very important. Golgi is adjusted, or it is present adjacent to endoplasmic reticulum. Okay, then if you check its phase, it has two phases. One has the cis phase and another one is the trans phase. Okay, the cis phase, yeah, cis phase will normally towards one side and the trans phase will be towards the another one side. Okay, so as you can see here, right, one side will be cis phase and another one will be, you can find the trans phase also. Okay, so how the cis phase will be and how the trans phase will be, as you can see. So we will see about that. Okay, we will learn about that also once again. This Golgi body structure, if you take about the structure, it also have the three different kinds of structures. One is the cisternae, and another one is the tubules, and another one is the vesicles. Another one is the vesicles. Okay, we have three different systems. One is the cisternae, another one is the tubules, and another one is the vesicles. All of them will form the Golgi complex. Okay, probably you can see here only, right, how the cis is there, how the trans is here. So here you can find cisternae. Okay, here you can find the cisternae, and here you can see the tubules, and you can also see the vesicles. So these are the structures as you can see in a regular basis. Okay. So 1 second, guys. We have certain other things today. So okay, we will do one thing. We'll stop our class here, and I'm just launching the poll. Okay. So what is lumen? Lumen is the empty space. So we will continue the class in the next class. Okay. Lumen is the empty space. Sorry, because nothing, because I already took one class, empty space. Okay. I completed another class, like, I don't know, like, I'm feeling like less energetic. So when I'm feeling less energetic, I felt myself like a little boring. That's why I thought of stopping the class and starting the class in the next, like, continuing the Golgi from the next class. Okay, nothing else. So lumen means empty space, nothing is there. Okay, like, sorry, nothing means something will be there, empty space will be there. Okay, that's called lumen. Any other doubts, guys? You can. Sperm has mitochondria where? Sperm have mitochondria in its body region. Okay, so So we divide into head and body and tail. Okay, head, body, and tail. Mitochondria will be normally situated in the body region. So if I write some, you can leave after the NPS. Okay. Sorry for discontinuing the class in between. Okay. That's what happened. Like I already took one class, right? I lost most of the energy there. That's the first class. That's all. So here we normally contain the mitochondria. Okay. These three structure in 3 dimensions. Actually, I will try to include one more image, electron microscopic image in the next class. So you can imagine the things. Okay. So I will try to include that. ER also helps in membrane genesis. Exactly. ER also help in the membrane genesis means the formation of membrane. That's what plasma membrane synthesis, all those things are same on. That's also there, Mohan. You got it. You got me. About the apoplast. Simple. Okay. Think about cells like this. Okay. And I will write yellow color as the plasmodesmata. Okay. Yellow color as the plasmodesmata. If the water move from here to here, okay, move from here to here through this way, this is the symplast because it's passing through the this particular plasmodesmata membrane. Okay, it may pass through from here to here through in this way without ever reaching the plasmodesmata. Just imagine it in a 3-dimensional layer. Okay, it may happen through something like this. It may happen through like this without ever reaching the plasmodesmata. Then you can call it as the apoplast. So symplast means through plasmodesmata. This is without the plasmodesmata. Okay. Bye. Sperm cells also contain all these cell organelles. No, sperm cells don't contain all these cell organelles. They only contain certain cell organelles which are needed. Like they contain lysosomes, mitochondria. Okay, but this endoplasmic reticulum and everything is reduced because there is no need of any more protein production or anything. That's why. Okay. So body knows inhibitors will restrict active transport, then still why it produce it? No, our body won't produce the inhibitors. There's some other organism will produce the inhibitors to for its defense. Like why snakes will produce the venom, because to get protection or to kill the prey, right? So that's how our body won't produce the inhibitors. What tail contains in the sperm? Tail contains the tubulin protein. We will flagella. We will learn that, right? That only it will contain tubulin proteins. So in apoplast, the molecules move into the inner intracellular space, or it also it will never move to this cell space. You are correct. It will move in the intracellular space. Okay, intracellular space. We will also once again that will come in the plant physiology. Okay, one more time, probably if you know. Okay. Flagellin protein. Tubulin. Flagellin protein. Tubulin, flagellin, all those kind of protein. Any more doubts, guys? Okay, guys. Then in YouTube, sorry for ending the class early. We will close the class. We will meet in the 26th. Okay, 25th we don't have any class. 25th is a holiday because of the Christmas. Okay. So happy Christmas, guys. I forgot. Happy Christmas.